Good morning, class. If you can hear me, please type in the public chat. You can hear my voice. You need to type in the public chat that you can and confirm, please. I can see quite a number of us have joined from Doreen, Kevin, Lewis, Jero, Moses. Please type in the public chat so that at least I can confirm that the audio is clear. Okay, thank you so much. Karibuni sana, Lucy, Caleb, Anastasia, Esther, Michael, Kipkoech, Naomi, Amos, Lawrence, Dennis, Vincent, Abigail, Michelle, Boniface. We are quite a number. Thank you very much. I think we are good to start. We'll be starting at exactly 7:30. And today we have quite an interesting session. We have an interesting session, and I would wish that we enjoy the learning today. Good morning, good morning. Please note that you need to join using your name and registration number. Sophia and Kada, and Judy Mweni, Amelia Nyawera, the same thing. Colonel, you join using your 2 names and your registration number, please. You join using your 2 names and the registration number. I want to believe we are people who can follow instructions. We are not people who don't know how to follow instructions. 19 pages. Remember, just to remind majority of you who are not joining the classes, before you sit for your cut on Monday, we'll check whether you meet the minimum class attendance. Coach, you need to join using your registration number. And I'll be checking from the YouTube chat how many have joined. So you need to meet the minimum attendance before you're allowed to sit for the cut. And And so good morning, and we can start now. I do believe that majority of us have joined. I can see from the charts we are quite a number. We are joining, and this is encouraging. And my name is Julius, and I'll be taking you through a very interesting topic. The topic is on the immune system, and then we will end up with checking or rather understanding the diseases associated with HIV and AIDS. But to start us with, we need now to know the definition of immunity, to understand the definition of immunity, what we mean by immunity. Secondly, we need to understand the tissues and the players in the immune system. Thirdly, we also need to understand the structural components of the HIV virus, and of course, be able to explain the clinical cause of the HIV infections, and of course, the accompanying symptoms. The accompanying symptoms that when you get infected, if you get infected, what are the accompanying symptoms and the disease progression. The session will be will involve around 1.5 hours. Will take around 1.5 hours, approximately 1.5 to 1 and 45 minutes. Again, to remind you, you are most welcome. I can see 217 are watching, and I encourage you to call up upon your colleagues. This is a very interesting topic. It's one of the most challenging in this course, and I have been tasked to take you through it. I do hope that we are going to enjoy the learning. I'll try as much as possible to be pausing and at least let you digest some of this content as we move on. Again, remember, I'm monitoring the public chat on the YouTube channel. So if you have anything that you wish that I clarify, you need to type in the public chat, and I'll capture it and I'll answer it to you. Okay, I'll answer it to you, but at least you need to type that in the public chat. So ladies and gentlemen, welcome to the class today. I think I've laid down the housekeeping rules, and to introduce us, number one, the very important aspect in this lecture is to know what is immunity, and the term immunity simply defines the defense. These are the defense system, just like we have the KDF, just like we have the national police in Kenya. Then immunity is all about the defense that is mounted by the body to infectious diseases. And some of you might be wondering, why do we need to defend ourselves? Remember, just like a country, just like an individual, you need people who love you, and you also need people who hate you. Most young people call them haters. And so even our bodies do have what is known as haters. The majority of these haters are infectious diseases. Infectious diseases arise from pathogens, microorganisms known as pathogens. I do hope that's clear. These are pathogens that take advantage of our bodies and cause disease. And then the second and quite an important point here is that the immunity simply refers to a system like I've shown you, quote unquote, simply a system of which constitutes of cells, different cells, of course, molecules, and even tissues. And so the most important thing to understand from this point here is that this is simply a collection of protective mechanisms, that help your body to fight diseases, to fight organisms that cause diseases. And so I would wish that you take it, and I have sent these course materials with you, so I believe it's very easy to understand and to fall from your end, that we are only looking and interested in the protective barriers, all right? And when we talk about the protective barriers, even the skin is part of the defense, because the skin is like a wall, just like every house have a wall. Even your skin is a wall. So again, we are interested in the organs that make this protection system. Again, we are also interested in the cells. And again, the other players are the molecules. So it's important that we be familiar with this, that immunity simply means a defense system that is mounted by the body. And this defense system is simply organized into molecules. Then of course cells to tissues, and tissues make organs. So it should be very clear what we mean by defense system. So ladies and gentlemen, next we familiarize ourselves with the general human body, all right? The general human body, and as you can see in this layout here, starting from the head all the way to your toes, you have the distribution of the immune system, starting from the head all the way to the toes. In the head, especially in the nose, you have a defense mechanism. These are known as the adenoids. And of course, around the mouth and the nose, we have the tonsils. If you move on inside, within the heart, we have what is known as subclavian vein, and another very important organ known as the thymus, this one here. It is placed at the top of the heart, all right? And then when you move on to the stomach, we have a spleen, all right? Within our armpits, we have the lymph nodes. In the small intestines, we have mechanisms known as the pious patches, and another appendage known as the appendix. Moving on down, we meet the lymphatics that you find in the legs. Again, in our groin region, in the groin region, you'll find a further collection of the lymphatic ducts or the lymph nodes. And of course, in the bones, this is where everything originates from. We have the bone marrow, So the bone marrow is where we get all the cells from the body, all right? So in a nutshell, this is simply the distribution of the tissues of the immune system. And as you can see, they are clearly and geographically positioned, starting from the head, the armpits, the general abdomen, of course, from the thorax. From the chest, we have the thymus, the spleen, the kidneys are also part of the immune system, the lymph nodes in the groin, and the lymphatics in the legs, and of course, the bone marrow. So this is generally the distribution of the defense system in the immune system, in your body right now as we speak. And so in the next slide then, we meet the prayers. In the next slide, we meet the prayers, and these are now in the blood. You find that you have a category of cells, and this category of cells is involved in the defense. We have the first category, and I would wish that you take note of this. The first category of cells is the phagocytes, which are further classified as granulocytes, and their broad name is the leukocytes. All of them are known as the leukocytes. This is on the left side. So we have those that have granules, like this one here, known as the neutrophil. The second category is the eosinophil, and the third category is the basophil. So all these three cells play a similar role in keeping off microorganisms and in keeping off pathogens from the body. And like I've mentioned, they are found in the blood, of course. Simply what you learned in high school to be the white blood cells, to be part of the white blood cells. Then, of course, we have the category known as phagocytes. the category known as phagocytes. A clear example is this type version of cell here, known as the monocyte. Then on the other end, on the right-hand side, you meet the category of cells known as the lymphocytes. And these are very important cells because they are the ones involved in majority of the body defenses. A group of cells known as the T cells, uppercase T and the uppercase B, and another one known as the natural killer cells or the NK cells. Then, of course, in the blood, we also have this category of cells that do not play any defense system, but help in other elements, like the platelets that aid in clotting of blood, and of course, the red blood cells that aid in the transport of oxygen. So all these cells, then, we find them in the blood, and of course, majority of them, with the exception of these two, they aid in defense system. Then moving on, in the tissues, you also find some cells that are involved in the defense system. And the first group of cells are the eosinophils. I think we have already mentioned them up here. So we have eosinophils in the blood and eosinophils in the tissues. Then there's a category of cells known as the mast cells. So these ones are most likely associated with allergic responses. Anytime you have been bitten by mosquito, anytime you have been bitten by a bee, these are the type of cells that are involved in allergic responses. All right, the mast cells. Then a very good category of cells known as the macrophages. And normally in common man's language, these are like the local watchmen that we find in our gate. The macrophages, they are the local watchmen that we find in our gate. Then, of course, we have another version of watchmen known as the dendritic cells. So this group of cells, the macrophages and the dendritic cells, are very closely related and play similar functions. Then, of course, we have the T lymphocytes also in the tissues. We have met them here, the T cells, and the plasma cells. The plasma cells, of course, they are associated with these cells known as the B cells. And then, of course, finally, we have the natural killer cells. So if you will take a pause, then I need you to type in the public chat. in the YouTube channel. How many versions of cells are you aware about? Okay. How many versions? Gatwiri, I can see you have a question. I'm going to answer it. Thank you for that question. I'm going to answer that question later. So I want you to type in the public chat how many versions of cells, of the immune cells, are you aware about? Okay, please type that in the public chat as we move on and as we take a pause. Do you know of any, or have you previously encountered any type of white blood cell, what we call white blood cells in the hospital? I need you to type the one that you know in the public chat. Type in the public chat in the YouTube channel the version of cell that you know. Good morning, Samuel Keno. Isaiah, teacher, I need you to name the cell, not sick cell. Don't name the number. I need you to name. I need you to name the type. I need the name, not the number. I need the name and not the number. I think you don't know how to follow instructions. I asked clearly, what cell do you know? And you are telling me 1 and 2 and 4 and 3. No, I need the name, please. Okay, Liz has gotten it. Cd4 cells, and Judith, lymphocytes. Gaitlin, CD4. Kevin says 4 cells. There's nothing like 4 cells. You are wrong. Maureen Bogo, you got it. T cells. Kidubi, granulocytes. Naomi Mudeu, white blood cells and red blood cells. Thank you. Abraham Muridi, you are wrong. I'm not talking about the number of cells. I'm talking about what cells do you know, or what cells are you conversant with of the immune system. Kipchoge says he's aware of the lymphocytes. Thank you. I think we are making good progress. Red blood cells are not, yes, red blood cells, of course, but their function is different. Very good, Isaiah Lotich. So we can move on. Please keep on typing the public chat. But before we move on, I need to clarify this. Someone has asked, where will Cat 1 cover? Cat 1 will cover up to what we have done today. Okay, I think I have answered that question. Cat 1 will cover up to what we have done today. All right, so that should be very clear to us, so that at least we are aware of what is expected for us in Cat 1. All right. Now, the next thing that I would wish to bring to your attention is why is it that whenever we hear about HIV, whenever we hear about anyone who is infected with HIV, we always hear the term Cd4 cells. We always hear the term Cd4 cells because I think this is a common misconception amongst people. Many people always hear HIV and then they hear Cd4 cells. So many people assume that HIV is the same as Cd4 cells. But Cd4 cells simply means a group. I need you to note that down. Cd4 cells simply means a group of T cells, maybe you can note that down, or known as helper cells, a group of T cells known as helper cells. So CD4 cells are a group of T cells known as helper cells, and these cells are very important because they are the target of the HIV virus. These cells are very important because they are the target of the HIV virus. And I'm going to be asking this question as we move on. As we move on before the end of the lecture, I'll need you to give me the definition. I'll need you to give me the definition of CD4 cells. Okay, as we move on, using your own understanding, I'll need you to at least tell me, what do you understand by CD4 cells? Remember, I have clearly mentioned that CD4 cells simply means a group of T cells, that are known as the helper T cells, and they are usually the target of the HIV virus. And what we mean by CD means it's not condom, because I hear most of you, when you see the word CD, you always, the implication in your head is condom. No, CD we simply refer to the cluster of differentiation. So by the end of this lecture, again, I'll also need you to define for me what we mean by CD, the cluster of differentiation. And at the end of the lecture, I'll also need you to type in the public chat what you understand by CD4 cells, okay? By CD4 cells. So we have Munyao Kamene who asked, where do T blood cell grow from, since they don't grow in the red blood cells? Munyao Kamene, we don't have anything known as T blood cell, okay? We have T cells. And as we move on in this lecture, I'll tell you where the cells come from. Do you understand? I'll tell you where the cells come from. Kimutai asks if there is any difference between CD4 cell and T helper cell. And I clearly said CD4 cells are the same as the T helper cells, Kimutai. Thank you for that clarification. All right. And I see 380 of us, or 379 are already streaming in. I expect more than 1,000 students to be streaming in. So remember, I'm checking the attendance, and I might remove you from the cut if you are not attending this class. I might say that you are not going to sit for the cut on Monday because on account of failure to attend the class. So let's move on, ladies and gentlemen. Let's move on. In the next section, then we need to define something known as innate and adaptive immunity. So anything known as innate means anything that you are born with, anything naturally acquired, naturally acquired from your parents when you are born. That's what we mean by innate. So this is a system of defense that involves what you acquired from your parents. all right? A system of defense that involves what you acquired from your parents. And what did you acquire from your parents? Number one, you are born with a skin, isn't it? Number two, you are born with noses and ears, and in your nose and ears we have mucus, and in your ears we have wax. You also find in your eyes there are some tears. Actually, the first thing that happened when you got born, you cried. Of course, you sucked milk from your mother's breast. Of course, there are some chemicals in the body, even in your stomach. All right. All these chemicals, all these systems are part of what is known as the innate immunity, what you are born with, okay? And these chemicals aid in the protection against infection, all right? Then the second part of immunity is known as the adaptive immunity, and these are the cells. These are the cells, and I have already introduced you to these cells. The cells known as the B cells, the cells known as the T cells, and molecules known as antibodies. Molecules known as antibodies. These molecules, they have a Y shape like that. They are known as antibodies. So all these three, the B cells, the T cells, and these molecules develop as you grow up. And that's why they adapt to your body. They adapt as an infant grows up. So this is what is referred to as the adaptive immunity. Adaptive immunity. So I do believe that it's clear what we mean by innate immunity and adaptive immunity. Remember the versions. Again, by the end of the lecture, I'll ask you, and you need to define for me, what are the components? What are the components of the innate immunity, and what are the components of the adaptive immunity? And by the end of this lecture, I need that answer in the public chat. I'll need the answer in the public chat. So Delaine Gasheri, this is not a greeting forum. You simply have to type the answers, okay? You simply have to type the answers. So we are done with that. So we are now familiar what we mean by the innate and the adaptive immunity. In the next slide now, we look at the structure of the HIV virus, the structure of the HIV virus. And we shall use this diagram here to understand the structure of the HIV virus. So simply on a shortened version, when we look at the structure of the HIV virus, number one, if you have to draw it, you start by drawing an S followed by another S, okay? And this S, these two S's are what is known as the RNA, the RNA. The RNA simply means ribonucleic acid. This is what I show here, the RNA, ribonucleic acid. After you draw that, then you draw small circles. You draw small circles, these ones here. That's an envelope there. You draw these small circles. You can try and sketch them down. Sketch them down. Sketch the small circles down. All right. Then inside we have a triangle, a cube, and a hexagon. A triangle, a cube, and a hexagon. And these are very important things. The cube represents an enzyme. The cube represents an enzyme known as reverse transcriptase. Reverse transcriptase. All right. The cube represents an enzyme known as reverse transcriptase. All right. The triangle, the two triangles, the orange, is it the orange or the brown? The orange triangles represent another enzyme known as integrase. Another enzyme known as integrase. Integrase, okay, it's part of the virus. Integrase is also part of the virus. And then, of course, finally, we have the hexagonal structures. They also represent another enzyme known as protease. Protease. So this should be very clear to us what we mean by that. Protease, it's an enzyme found within the virus. So by the end of this lecture, again, I need you to remind me by the end of this lecture, I need you to type in the public chat, how many enzymes do we find in the structure of the HIV virus? We have clearly mentioned the first enzyme, which looks like a cube, is the reverse transcriptase. The second one, which looks like a triangle, is the integrase enzyme. And the third one is the protease, which looks like the hexagon. All right. Then, moving on, you find another envelope here. The pink, is it the pink or the purple one? The purple one. All right, the purple envelope, which are known as the matrix proteins. The matrix proteins, and they are usually abbreviated P17. P17. Then on top of that, we have the dark blue. We have the dark blue. Again, that forms part of the matrix proteins. Then we have these structures that look like a hammer. The top part is known as the envelope proteins. The envelope proteins, also abbreviated as glycoprotein 120 or the GP120. And the harder part of the hammer is known as the glycoprotein 41. The glycoprotein 41. So the top part of the hammer, or the hammer itself, is the glycoprotein 120, and the handle is the glycoprotein 41. The handle is the glycoprotein 41. So I mentioned we have this purple structure, the core proteins, and of course we have that part of the hammer and that part of the hammer, and that simply makes the structure of the virus. That simply makes the structure of the HIV virus. And remember, this envelope here consists of such a molecule. It consists of such a molecule, and this molecule have such structures. These are known as the heads. Okay, these parts here are the heads, and these parts here are the tails. These parts here are the tails. The head and the tails. So in a nutshell, this is the structure of the HIV virus. So by the end of this lesson number one, I also need you to tell me that besides the number of enzymes that we find in the HIV virus, I need you to mention for me how many proteins can you name in the HIV virus? How many proteins can you name in the HIV virus? All right. So we have encountered 2 or 3 proteins. So I need you to remind me and type in the public chat. You type in the public chat in the YouTube channel the number of proteins that you can recognize in the HIV virus. The number of proteins that you can recognize in the HIV virus. Please do that quickly. Please do that quickly as we move on. Please do that quickly. Now. In the next part now, I show you how does the virus interact. How does the virus interact with the body cells? And what is most important here is we need to appreciate that virus interacts with cells that have molecules on their surface known as the Cd4 receptors. So meaning the cells of the body, the T cells, okay, this is a T cell, they have receptors. A receptor is an antennae, okay? A receptor is an antennae. So this antennae forms the binding site of the HIV virus. The antennae looks something like that. Okay, so that is the antennae, and this antennae form part of the attachment. So this is a T cell, and of course the virus binds to this antennae. Okay, and the antennae are what is known as the cluster differentiation for receptors. We have a number of cells having this cluster differentiation for receptors. The first category is the T lymphocytes, and I already clearly told you these T lymphocytes are known as the T helper cells, followed by another category of cells known as the macrophages, the monocytes, and the dendritic cells. So again, Another assignment by the end of this lecture, we need to appreciate, or we need to name, you need to name for me 4 cells that have CD4 receptors. You name for me any 4 cells that have CD4 receptors. Okay? So this is the cells. These are the cells that are targeted by the HIV virus. So remember, one important aspect of viruses, one important aspect of this virus. So this is the structure of the virus. So a virus, normally majority of the virus, they do not live on their own. Okay, like viruses do not exist alone. Viruses do not exist alone. So it's very important that we already are aware about that. Viruses don't exist alone. They are like parasites. Okay, they are parasites. They need cells to survive and to multiply. Okay, so viruses are simply parasites. On their own, they do not exist. They only exist in this form, an RNA. An RNA, like I told you, it simply means it's ribonucleic acid. Ribonucleic acid. So these are nucleotides, nucleic acid. So, another example. Please, okay. Lazima yuko. Najua wapi. Yes, walikuwa Nairobi. Enda nayo mtakuja. Nitakuja kukuambia chenye tutafanya. For the wewe enda nayo hakuna shida. Patia kadipo. So I have clearly mentioned, sorry for that. I have clearly mentioned that RNA simply means ribonucleic acids. Ribonucleic acids, and these are simply a sequence of nucleotides, closely related to what you have heard about DNA. Okay, so this is a sister or a cousin of DNA. RNA is simply a sister or a cousin of DNA. So the most unique thing about viruses is, number one, they do not exist on their own, and number two, they have to exist inside a host cell. And that's why I clearly told you the analogy that they probably are more parasitic. They cannot exist on their own. They need another cell so that they can multiply and they can exist. All right. So now we move on to the next slide, whereby we ask ourselves then, how does the virus infect the cells and how does the virus infect the body? Okay. How does the virus infect the cell and how does it infect the body? We are going to explain that using this diagram here. This diagram here. So here we will meet this the virus, what is known as the virion. Whenever you encounter this, it simply means the virion. All right. So the virus infects a cell that has the CD4 receptor. So down here, this is the cell. Okay. All this is that cell. So the virus attaches to this cell on the CD4 receptor. After that, the membrane of the virus and the membrane of the cell fuses. Here, the membrane of the virus and the membrane of the cell fuses, and that ends up transmitting the content of the virus. I told you the yellow parts in the virus are the RNA. So they are transmitted into the cell. And then inside the virus, we met an enzyme that we referred to as the reverse transcriptase. So this enzyme changes the RNA, this one, into DNA. Normally, RNA has a single strand, as you can see clearly. RNA has a single strand, and it is changed by this enzyme into a double-stranded DNA. And then that DNA joins the DNA of the cell. It joins the DNA of the cell, and ends up making more RNAs. So these RNAs are transmitted outside. And they make another virus particle here. So the RNAs here, they join with HIV proteins. They make a new version of this virus particle. So in so doing, because we have interfered with the DNA of the cell, this cell will die. And when this cell dies, and we have already made a new virus or two new viruses, they will infect another two cells. Okay, they infect another new 2 cells. So clearly, in a nutshell, we move on from the HIV virus attaching to the new cell. All right, the membrane of the 2 cells fuse. Okay, after the membrane of the 2 cells fuse, the virus empties its content into the cell, and the content of the virus is the RNA, the single-stranded RNA, and the enzyme reverse transcriptase makes a double-stranded DNA, which joins the cell, the host DNA, and then from that they make a new version of the RNA, and this new version of RNA is assembled into a protein, and then the protein coat is assembled into a new virus. We call them the budding of the new viruses. And so we end up making 2 new viruses from 1 new viruses. The outcome, the end result of this process is that this cell, the whole of this cell will die. Why? Because its DNA has been interfered with. So that is simply, in a nutshell, the life cycle of the HIV virus within the cells. So maybe we take a pause and ask ourselves, which are the 4 cells that are infected by the HIV virus? I need you to type that in the public chat. Name the 4 cells that are infected by the HIV virus. Of course, I'll start by helping you. We say the first one is the T cell, but this T cell is which one? So can you please type that in the public chat? And I can see someone asking a very important question. What is the difference between DNA and RNA? So the difference between DNA and RNA, as you can clearly see in this diagram, an RNA is single-stranded and a DNA is double-stranded. We have a single strand for the DNA, for the RNA that is, and single strands for the DNA. I hope I've answered your question, John. I say don't use such names. John Cook guy, don't use such names. Use your name and registration number. Okay? Use your name and registration number. So I hope I'm getting my answers in the public chat. Cd4 cells, we have Cd4 cells, T lymphocytes, macrophages, and monocytes and dendritic cells. Thank you very much. Then we also mentioned that besides the Cd4 cells, besides the Cd4 receptors, we have other receptors in the cells that are known as they are simply abbreviated as the CXCR4. Chemokine chemoreceptor 5 and 4 that are also found in these cells, and these also are used by the T cells to bind, used by the virus, to bind to the T cells. So we have the major types. I think I have clearly given you that here. The major types that are infected by the HIV virus, the T lymphocytes, the T helper, the macrophages, and the dendritic cells. The macrophages and the dendritic cells. All right. So I think I've clearly explained that. That should be very clear how the virus infects cells. Now, in the next slide, we then ask ourselves, after infection, how does the disease progress? How does the progression of the acquired immunodeficiency syndrome progress? And the first phenomena we need to understand is that this takes time. It really takes a lot of time. Sometimes it can take as many years as 10 or even as 20, or it can even take months or days, because it depends with an individual and the rate at which the cells will be destroyed. The rate at which the cells will be destroyed. And usually what happens, like we have seen in the diagram, the T cells get, depleted. They get destroyed slowly by slowly. And when cells are destroyed, we call that a cytopathic effect. That is known as a cytopathic effect. And it results in the depletion. It results in the reduction of these cells. And when these cells are reduced and they are supposed to be helping the body in protecting it against infections, then it means that the disease progresses very fast. The disease usually progresses very fast. So normally, during the primary infection, the Cd4 cells get infected, or the dendritic cell gets infected. And of course, these cells are transported to the lymph nodes. I said clearly the lymph nodes are found in the groin and in the armpit. The lymph nodes, you find them in the groin and in the armpit, okay? That's why when you have a wound in the arm, you always feel some pain in the armpit and in the groin. So whenever they are transported to these two regions, then the virus continue multiplying and spreading throughout the body, until you get what is known as latency. Sometimes the body is able to control. The multiplication of the virus. And of course, when the body is not able to control the multiplication of the virus, we end up destroying a lot of T cells. And the outcome of that is the acquired immunodeficiency syndrome. So if we go back here, number one, we start with the infection that we have clearly demonstrated. Number two, the spread. And at this juncture, if we take your blood and check for the virus, we can be able to detect it. So this is what is referred to as viremia. Then, of course, after that, there is the immune response. The immune response is now the body trying to respond to control the replication of the virus, to control the multiplication of the virus. And that, if the body is successful, then that ends up in latency. So the phenomena of latency simply implies that the body is in control. It has arrested the multiplication of this virus. If that fails, if and when that fails, all right, if and when the immune response fails, then you end up with the destruction of T cells, and not just destruction, even depletion. They go low, all right, they are depleted. The actual amount of T cells go low, and you end up with a full-blown acquired immunodeficiency syndrome. So I do hope that's clear, and we can take a pause. And when we take a pause now, you can type it. I'll be checking the public chat whether you are, how many of us are able to answer my questions. Number one, I needed the definition of the Cd4 or the CD. The definition of the CD. I also needed the definition of the. I also needed the components of the enzymes, rather, not the components. The enzymes that we find. The enzymes that we find in the HIV virus. The HIV virus. I also needed you to name. I needed you to name the proteins. The proteins that are found in the HIV virus, and I also needed you to name. You have to name what? You have to name the four cells, the four group of cells that are infected by the HIV virus. You need to name that, the four group of cells infected by the HIV virus. Please do that in that order. The enzymes, the proteins, and the four group of cells infected by the HIV virus. All right. So we take a 1 minute breather. You sip some water and then we proceed. I don't have water. So Perez Cherotich, there's nothing known as intergrade. That's wrong. There's no enzyme known as reverse intergrade. Please type and type properly. Very good. I can see majority of us are attempting. Newton Mwega, we don't have proteins. I need you to name proteins, not proteins. Very good. I see majority of us are really following. CD means cluster of differentiation. Francis and Wycliffe Kimtai, thank you. Yep, Chepkolil Karen, that's a very good definition. Brian Kagai, we don't have a protein known as proteins. Name the proper protein, no, not proteins, please. I am following, and of course, I'm going to provide a feedback on this in the e-learning platform for all those who have been attending and those who are able to respond to these queries. This is part of the learning and it forms part of the assignment. And remember, the assignments are marked out of 10. The assignments are marked out of 10. So take it with the seriousness that it deserves. Right, then we proceed with Jeff Ishma. Why can't Al? What do you want? Please take your time to type. Don't just type nonsense. You type something that makes sense. So now, next, ladies and gentlemen, we look at viral load, what we mean by viral load, and this is simply a test. This is simply a test for measuring the number of the viral particles per milliliter of blood. So this is normally what is applied in the clinical settings. So in the clinical settings, then we detect the number of the viruses. We call them copies. Okay, we call them copies, and this simply notifies the clinician if a patient needs to start treatment, or if the patient can delay treatment, and what medical advice can a patient be given. But what happens after on exposure to HIV infection? Perhaps you have been exposed to the HIV virus. So within a very short period of about 2 to 4 weeks, within that period, then the virus intensely, multiplies within your blood because it keeps on destroying these cells. And then, of course, it reaches a point whereby the body is able to control. The body is able to control this replication, and then that results will now result in what we call the latent state, whereby the body is able to arrest the multiplication of the virus. But within the very first 2 to 4 weeks, then the rate of multiplication of the virus is usually very high. And normally, we do use these cells known as the Cd4 cells. We do use these cells known as the Cd4 cells to, inform us on how the virus is able to multiply. Because, like I have mentioned clearly, these cells are the main target. They are the main target of the HIV virus. So if you are able to know what is the normal count of these cells, then you can compare that and you can tell the progression of the virus. So this is now what is now used as a clinical marker to determine how fast the disease is progressing, And most importantly, if the disease or the virus is not arrested by the immune system, if your immune system is not as strong enough, then this phenomena may arise the immune depression, whereby the CD4 cells are quite depleted. They get damaged. We say they are reduced and they are impaired in their functions. So this now is what will results in the full-blown clinical development of acquired immunodeficiency syndrome, the AIDS immunodeficiency syndrome. For the detection, ladies and gentlemen, normally for the detection of the virus, we have a specific type of a test that is used. It's called ELISA. Majority of us might have come across it, ELISA, and simply ELISA means enzyme-linked immunosorbent assay. So ELISA is simply for testing the enzymes, and the enzymes are simply proteins that we find in the viruses. They are simply proteins that we find in the viruses. But then there is a period during which the body starts producing antibodies. The period during which the body will start producing antibody and is able to fend off the virus is now, that is a phenomenon of the state of seroconversion. And this will take about, after the 2 weeks, it will take about the 25 days. It will take about 25 days. And immediately after infection, the virus cannot even be detected in the blood. The virus cannot be detected in the blood. And we have said clearly, depending on your immune system status, if and when the body is able to arrest the virus, then that will end up in the latency. And the latency means that the virus is kept in check. And when the virus is kept in check, then the implication is that then you can stay with this virus for as long as you are not immune compromised, as long as your immune system, your defense system is not compromised. That's the implication of the asymptomatic stage. Asymptomatic stage. We have individuals or other people who, when they get infected, they may stay for a very long time with the virus without developing the actual full-blown acquired immunodeficiency syndrome. So these are the people referred to as the long-term non-progressors. The long-term non-progressors simply means you are HIV positive, you have the virus within you, but you don't have any symptoms. No symptoms whatsoever. No symptoms whatsoever. And these are the people who have the very high risk. They have a very high risk of now spreading the virus. So such a person is always encouraged to keep on getting checked. And this is why we are always encouraged to always constantly keep on getting tested to know our statuses. Now, the danger is you have the virus, you are immunocompromised, and your body cannot fend off the virus. The production of the CD4 cells cannot match the destruction. The production of the cells that are getting destroyed cannot match the destruction. So it means that then because these cells are quite important in keeping off, general infections, it means then you will get these kind of infections. You find that every now and then you get some funny infections like urinary tract infections, you get pneumonia, you get some coughing, you get some tumors, some wasting, you are simply wasting, you are simply sweating excessively, and you cannot tell the reason why this is happening. And so this is now what leads to clinical disease, the clinical disease of the acquired immunodeficiency syndrome, which has several phases, starting with the acute phase that is characterized by the fevers. You sweat excessively, you have sore throats now and then, you have malaise, you have swollen lymph nodes, etc., etc. So during that phase, then it means that the multiplication of the virus is quite high. And then, like we have seen in the diagram up there, if and when you boost your immune response by proper eating, by taking the proper medication for the opportunistic infections, then the body can result into latency, or rather being able to fend off or to arrest the multiplication of the virus. But in the absence of that, then you have the clinical full-blown acquired immunodeficiency syndrome, acquired clinical immunodeficiency syndrome. And so this is what happens in a nutshell. In a nutshell, we can clearly see what happens. You are infected at this day 0, all right? At the point of infection, you have quite a higher number of T cells. So the y-axis here shows the T cells per millimeter cubic of blood, per 1 millimeter cubic of blood. They are usually very high, over 1,000 in a healthy individual. But remember, this is quote unquote healthy individual. So if you get infected here, this is the virus, and these are the number of T cells, the number of the so-called Cd4 cells. So if you get infected within 0 and 3 days, because the virus starts multiplying very fast, then as you can see, the opposite is true for the Cd4 cells. They start dropping, and they drop to a low of allowed 500 cells per millimeter cubic of blood. And then, of course, at that juncture, maybe within 1 or 2 weeks, maybe the body is going to wake up and start feeding the virus, arresting the virus replication. And that may arrest the replication. But then within years, now this was weeks, the first part here is weeks, but then within the first, second, fourth, all the way to 11th year, then the virus might be able, because your body is also growing weak, the virus might also be able to continue multiplying, and the Cd4 cells continue getting destroyed to a level whereby they near 0. So this is a very dangerous state where you can find someone getting infected with so many infections, from tuberculosis to pneumonia to diarrhea to anything that can infect an individual. So normally this part is the dangerous part that constitutes all these symptoms and the clinical latency. The clinical latency whereby the body is able to, arrest the virus. But it's also very dangerous in the sense that if you get immunocompromised at any juncture, then the virus will continue multiplying, and at this point, opportunistic diseases set in. And at this point, when the virus is at this level, then the patient may die. The patient may die. So this is what we use for understanding the clinical cause of the HIV virus disease, how the clinical cause of the HIV virus disease progress, and what is expected at what juncture. And this is why we said the reason why these cells, the cluster differentiation 4, are used for detection of the infection is because they represent the vice versa of the virus. When they are high, the virus is low. When they are low, the virus is high. Okay? When they are high, when the CD4 cells are high, the virus is low. When they are low, like at this point here, the virus is quite high. All right? So normally they are used as markers for the detection of the detection of the infection, for the detection of the infection. Now, of course, like we have mentioned in previously and in class, the disease is not as dangerous as it seems. All right, because due to advancement in treatment, due to advancement in vaccination, you can control the progression of the disease using therapy, using medications, drugs that target to destroy the virus. They are called antiretroviral drugs. And normally there is a therapeutic approach. Patients are given a highly active antiretroviral therapy or a combination antiretroviral therapy. So this therapy tries to help and keep the virus level low. And again, the therapy also involves trying to control other opportunistic infections like sarcoma, lymphoma, and other complications. Okay, and this therapy, these drugs, they inhibit the enzymes. They inhibit the enzymes that we found in the virus. The first enzyme we found in the virus was transcriptase. There was another enzyme that we called protease, and we also had another enzyme known as integrase enzymes. So these are this is a function or the mode of mechanism of functioning of antiretroviral drugs. Antiretroviral drugs. So now, at this juncture, I need you to type for me in the public chat. What is your take-home message? Number one, can you define what is immunity? Because we need to look at the summary of the first part of the lecture before we start the second part of the lecture. We need to take 5 minutes break, then we start the second part of the lecture. Can you define immunity? Number one, I need you to define immunity in the public chat. Okay? That's number one. The first assignment that we are doing in the public chat, define immunity as you understand it. Number two, name the tissues of the immune system. We are doing a summary and we are noting it. Yes, in the public chat. Anthony Mwangi, I am following whatever you are doing, and I need that named in the public chat. Right? Purity Mwende, all these things are in your email. I have sent them in your email, and they are quite there. Even in the e-learning platform, I have updated everything. Number three, explain the name the cells that have CD4 receptors. You name the cells that have CD4 receptors. We are taking a 5-minute break, and in the 5 minutes break, I need you to complete that assignment. And number four, I think you can first of all finish that. We take a 5 minutes break, then we do the second and the last part of the lecture. And I know that some of you are complaining that some of these notes are not updated in the e-learning platform. They are. I have updated them today in the morning, and and other than that, I have also sent them to you in your emails. So can you open your school email and you are going to find that. We take a 5 minutes break and then we start the second part of the lecture. We take this the second part of the lecture. And you're also going to find some questions which you are supposed to attempt in the e-learning platform, in the discussion forum in the e-learning platform. We quickly do that and start the class. We proceed at 8:38. We take a 5 minutes break. We do we answer those questions and then we proceed at 8:38. Okay, we come back to class now. I think we are doing very well, those of us who are attending, and thank you so much for attempting the assignments. I can see we are learning. Learning you don't have to suffer to learn. You need to enjoy to learn. So ladies and gentlemen, we've done 1 hour and maybe allowed 10 minutes, or about 1 hour and allowed 15 minutes even. And so in the second and the last part of the lecture, I'll take you through the HIV comorbidities. Sometimes I have a problem pronouncing this word, comorbidity, and it depends with where I was not born, all right, and where I did not grow up. And because nobody chooses where you grow up and nobody chooses where you are born, then you have to bear with my pronunciation. I hear most of you call it Chida Matamchi. Now, ladies and gentlemen, this second part of the lecture introduces us to something that we are already familiar with. We've clearly said that the virus known as the HIV causes a wide variety of diseases, and this wide variety of diseases is what is known as the Acquired Immunodeficiency Syndrome, AIDS, all right? So the term comorbidity simply refers to when you have more than one illness. You know, you are suffering from common cold, and you're also suffering from headache. You're suffering from tuberculosis, and you're also suffering from typhoid, okay? So comorbidity simply is the act when, or a disease or a medical condition that, you know, is present. Two medical conditions are present at the same time within the same individual. All right, within the same individual. And previously, if we go back to the lecture, we've seen that whenever you get infected with the virus, then at some point you are going to be infected because of the low immunity. You are going to be infected by what we have just previously referred to as opportunistic infections. You know, you find an individual suffering from, you know, two types of infections that are not closely related. All right. So this is the phenomena of comorbidity. So number one, we should be able to define what is a comorbidity and how does a comorbidity arise. Okay. Number two, we should be able to explain the common comorbidities associated with HIV, with the infection with the HIV viruses. The common comorbidities are associated with HIV viruses. And without preempting much, the first thing that I would like you to understand is anyone infected with HIV are prone to diabetes. They are also prone to a condition known as hepatitis C. They are also prone to hypertension, or what you people refer to as high blood pressure. They are prone to kidney diseases. They are prone to mental illnesses like depression, and they are prone to substance abuse, kuchoma, especially kuchoma, because I know some of you are quite good in this, all right? And ulevi, ulevi na kuchoma. These individuals are prone to all these conditions. And anyone living with the virus is referred to as a person living with HIV/AIDS. All right, anyone living with the virus is classified, simply categorized as a person living with the virus, or PLHA. Whenever you come across that term, it's the term, okay, well, not in a derogatory way, not in a very discriminative way, but this is a simply group of individuals who are living with the virus. So this is not a term used to stigmatize these people. No, it's only a term used to help identify them and identify the common comorbidities that they may be associated with, which I've just listed for you. Okay, so you find that other than that, there are higher chances of the liver diseases and the renal diseases, the kidney diseases. All right, And then other than that, we have very high chances of the STIs, the sexually transmitted infections, like this one that causes cervical cancer, the herpes simplex, this one that causes syphilis, the gonorrhea, and the mycoplasma, all right? The worst part to these individuals, especially those who reject their conditions, those who do not accept that they are infected, they suffer from psychiatric disorders, anxiety, depression, schizophrenia, and you name them. And due to the sensitization, of course, nowadays we have accepted. In fact, in my class on Mondays, those of us who meet at LT01, and anyone is welcome to meet with us at LT01, we have discussed and mentioned that this disease is not even as dangerous as we think. We have other dangerous diseases, like currently the epidemic that we have, the COVID-19. We also have, the menace of cancer. If you get infected with cancer right now, if you get the cancer, chances are high that you are going to die. But if you get infected with the HIV viruses, you might even have a longer life than someone who have already been infected with the cancer. And to make matters worse, majority, something like cancer, depending on which type of cancer it is, it's not even an infectious disease like HIV. It's something that is in your genetics, all right? So the most important thing here, the point I'm trying to bring to you is that some of us, even in our class, were born with this virus. It's not your own mistake. If you simply have an attitude change, if you simply adopt the drugs that are given, and they are given for free to these individuals, and if you simply accept and have a behavior change, then you are bound to live longer than even those of us who have not contracted the virus. So it should be as clear as day and night that getting the virus is not an arrest. You are not arrested and put in prison. No. All right. You have, number one, to accept who you are. And I've even seen individuals in TikTok who have embraced their status. They even use TikTok videos to encourage others that you should always be taking medications. I'm taking medications. You see an individual in those videos taking the antiretroviral drugs and mentioning that I was born with this virus. What am I supposed to do? Some of us who get infected, it's not your own fault. It simply did not mean that you are promiscuous or anything like that. Nope. All right. So in case, and even you, an infected individual, you should not stigmatize anyone. You should not stigmatize anyone if you know that someone already is living in that situation. You might even be having you might even die earlier than this person, if you, especially under unfortunate circumstances, if you get to have to contract any type of cancer or tumor, for that matter. Then, of course, for individuals living with conditions like the blood sugar, diabetes, then they need to keep on monitoring. If you have any kidney illnesses, then you need to go for dialysis. And as you go for dialysis, then you should always inform the medics about your conditions. All right. If you have high blood pressure, again, all these things you only need to monitor. And when you monitor and take the correct drugs and the diet, you avoid smoking and you do exercise, then you can live longer. Same case with hepatitis C. The problem with this one is that it can lead to some cancer, especially for cervical cancer. But the good thing is most of it can be vaccinated, and not only vaccinated, but it can also be treated. It's curable. Same case with depression, same case with mental illnesses, and same case with drugs and substance abuse. So all these things you only need to know, where am I? And avoid the so-called club drugs, because I believe majority of us are in this group. They take the club drugs, Avoid all this at any possible cost, all right? At any possible cost. So the take-home message from this section 2, or rather second part of our lecture today, is you have low chances, minimal chances of dying from the virus, okay? You have minimal chances of dying from the virus. but what may kill you are the complications of the disease. The complications of the disease. And these complications arising from the disease are what we have just called the comorbidities. And the good thing, majority of the comorbidities can be controlled. They are treatable, they are curable, and they can also be handled and properly managed. So it's only up to you to be able to know where you are. All right. Then in the third and the last part of this series of the lecture, we ask ourselves, what are the recent advances and the challenges in the cure of this disease? The challenges and the advances in the cure of this disease. And I'll refer you to the e-learning platform. In the e-learning platform, and I'll refer you to topic number 6. Topic number 6, all right, in the e-learning platform. That's where I am right now. Topic number 6. I have created a discussion group. So I need you to, number 1, listen to the videos, the YouTube videos that I have attached there, and then you do a summary. You do a summary using your own words, okay? You do a summary using your own words. Topic number 6 on immunity and the HIV. I have that's an assignment, and the deadline, of course, is indicated even there. It's on 26th. The deadline for submitting that assignment is on 26th. All right, but then currently we do have too many people that have high chances of getting infected. But then the challenge is that the access to prevention, care, and treatment, and there's still no cure. The second challenge is that the virus does not just affect healthy individuals; it impacts everybody. It impacts the breadwinners in the households. It impacts communities. It impacts anyone. All right, and of course, the worst hindrance to management of this disease is stigma and discrimination. The stigma and discrimination, especially from those of us who think that we are not infected, and especially from those of us who think others are infected, and they do not know they are also living in danger of getting infected. So having said that, it's up to you. I have told you that getting infected with the disease is not a life imprisonment, okay? It's not a life. There are even more dangerous things in this life than this virus as we see it. So this issue of pointing fingers to people who are living with the virus should cease, and even remember, you have higher chances of dying from COVID-19, the new virus known as COVID-19, than dying from the HIV virus. You can add more, and at that juncture, I would wish that we stop there. And we welcome any remarks, any questions in the e-learning platform. I'll try to answer in the next 15 or 10 minutes. I'll try to answer any questions before we can I can allow you to go to your respective classes and meet your respective lecturers. So let me see what I can, which questions I can answer to in the YouTube channel. So maybe before we leave, please define what is a comorbidity. Define what is a comorbidity, and name 5 comorbidities arising from HIV infections. You do that in the public chat in YouTube channel. You do that in the public chat in the YouTube channel. Okay, please do that quickly. Define what is a comorbidity and name 5 comorbidities associated with HIV. Name 5 comorbidities associated with HIV in the public channel. Steve Odongo asks, how will you submit the assignment? Steve Odongo, the assignment is to be submitted in the e-learning platform. There is a discussion group that you are supposed to join. And to everyone else, everything is done in the e-learning platform. You do that assignment in the discussion group. You create a discussion group and give me the answers in that discussion group. So the last assignment, we are doing it in the e-learning channel. I mean, in the YouTube, in the YouTube comment section, you define a comorbidity and also name 5 comorbidities. Braham Muridi, you move to topic 6 of the e-learning channel and click the discussion, and that's where you are supposed to submit the assignment. Most of us do not know how to follow instructions. Branham Muridi, again, you open the e-learning channel. Topic 6, there is a discussion group. Ay, 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 ay. Naftali Balu, you are typing in German. I cannot understand. Danke, I understand is thank you. But for Ikition, I don't understand that. You type in English. That is our medium of our communication. You are defining comorbidity, and then of course you list 5 comorbidity. Lucky, you are sleeping. You ask where is the assignment, and I clearly keep on mentioning, repeating the same instructions. I will throw you out of my class. You are sleeping. Okay, we have 5 minutes to finish doing that assignment quickly. 5 minutes, then at 9 we go to our respective venues and meet our respective lecturers. Hello? All right, we have done 1.5 hours. I can see the response is very good in the charts. We are able to define what is a comorbidity. We are able to list the at least 4 comorbidities. That's good, encouraging. Right, so I see it's 9 a.m. right now. You can log out and then you move to your respective venues to meet your lecturers. Thank you very much for participation, for active participation, and for showing up in class. For those of you who have not been able to attend the class, I encourage them that these classes are recorded, and you can always find the proceedings in the e-learning platform and also in the YouTube channel for follow-up. If you have any questions, you type in the e-learning platform, and I'm able to follow up with you. Have a good day, and thank you very much. Please log out. Log out now.